Cell division. So in body cells, chromosomes are found in pairs, and body cells divide through a series of stages called the cell cycle. During the cell cycle, the genetic material is doubled, then it divides into two identical nuclei in a process called mitosis. Before a cell can divide, it needs to grow, replicate DNA to form two copies of each chromosome, and increase the number of subcellular structures. So that forms stage 1. In mitosis, one set of chromosomes is pulled to each end of the cell, and the nucleus divides. That forms stage 2. And finally, the cytoplasm and the cell membranes divide to form two identical cells. That forms stage 3. Mitotic cell division is really important for multicellular organisms because mitosis and this form of cell division allow an organism to grow and to repair itself of any damage. So mitosis is important for growth and repair. So let's look at growth and differentiation then. So in animals, first of all, in animals, most differentiation happens at a very early stage in development. By the time a baby is born, it already has all of its cells differentiated and specialised to form specific functions, and a baby is born with that already having happened. There are very few cells within that animal that continue to be able to differentiate into other things after that point in time, and those are called adult stem cells, and we'll look more at those later. But most of the cells in that body, the differentiation will have happened and they will already have their role to play. In plants, however, we see a different system. Plants, many types of cell, have the ability to differentiate throughout life. It doesn't stop at the point of birth as with animals, so plants can continue to differentiate throughout. And that differentiation mainly happens in root tips and in meristems, and you can see here an example on the diagram here of your root tip, where that mitosis continues throughout the life of the plant, that growth continues throughout the life of the plant. However, other plant cells still have the ability to differentiate, and that is why we can take cuttings from plants and create whole new plants, because you can take a cutting from a stem of a plant or even a leaf with some plants, and those cells are still able to differentiate as that plant continues to grow and form a whole new set of differentiated cells to form a whole new plant. Okay, let's look at stem cells then, first of all, in animals. So in animals, they come in two forms. There are embryonic stem cells and there are adult stem cells. Embryonic, we are talking about human embryos. So if it continued to grow, it has the potential to become a baby. Okay, so we're talking human embryos, and you can see there are probably going to be issues with that, and we'll talk about that in the next slide. Adult stem cells are found in the bone marrow. The key thing here is obviously embryonic cells are actually more useful to us than bone marrow cells and adult cells. At the point of the cell being embryonic, it still has the ability to differentiate into many different types of cell, because that embryo is still growing to form all the body parts that it needs to function. So human embryos and embryonic stem cells are very valuable in terms of their ability to differentiate into many forms. Whereas adult stem cells in the bone marrow have a limited amount of cells they can differentiate into. So they have some use, but not as much as embryonic cells. Why are these stem cells important to us then? And you can see on the diagram at the side there, Medically, embryonic stem cells can be removed. They can be cultured. Those stem cells can then be cultured to form specific cells that are needed to potentially replace damaged cells within the human body. So there is hope that they could be used for treatment with things such as paralysis, where nerves have become damaged and they could be used to replace damaged nerves, or with diabetes, where body cells have stopped functioning properly within the blood glucose system, and they could be replaced within the pancreas to work properly as well. You can see here there's also other suggestions for heart and kidney. There are many possibilities for stem cell use to cure or aid in the recovery from conditions and accident damage. Plants then, having a think about plants as the other version. Now obviously we've said plants can do this pretty much their whole life through, and those mainly are found in the meristems. Okay, so the tips of the roots, the tips of the shoots, where most of that growth is going to happen within a plant. So how are stem cells useful in plants then? Well, this means that we can make copies, we can clone a plant very quickly, and we can make many copies very quickly. Now, why is that useful? Well, if we've got a plant that is valuable to us, so let's say we've got a tomato plant that produces a lot of fruit. If we can make identical copies of that exact plant, we suddenly have many plants that can create that exact amount of fruit. So financially, it's incredibly valuable for the farming and the agricultural industries to be able to produce crops that are very useful to them, and then clone and replicate that very useful crop. Allowing also for things like research in plants, because we can make many copies and we can look at the way diseases affect plants, for example, and things like that. So very useful for agriculture, horticulture, and research. Okay, let's think about some of the issues then associated with stem cells. So firstly, when we're talking about embryonic stem cells, we are talking about getting embryos either from aborted pregnancies or spares from fertility treatment. Okay. There is research at the moment going into something called therapeutic cloning, whereby an embryo would be cloned and created from the patient's genetics. The reason for this is that if the stem cells are genetically similar or identical to the patient, there is less likelihood of the body rejecting those cells. So that is another thing that is being looked at the moment. What are the issues that we've got with this then? Well, obviously, first of all, it's where the embryos are coming from. Number of issues associated there. We have ethical and religious aspects to consider. Okay, firstly, is it okay to take these embryos? Because at the end of the day, they are potential life. If those embryos continue to grow, they would potentially be a baby and a human in their own right. And obviously there are conditions and issues with the fact that that embryo doesn't have human rights, in a sense that it can't speak for itself as to whether or not that's acceptable usage. Then we've got the sort of religious point of it, and this varies across religions specifically, but the general principle being that you're interfering with a natural process. You're interfering with nature and with natural reproduction. So there are issues with the fact we are interfering with the reproductive process and interfering with nature in that sense. One other thing to think about, and I said before, adult stem cells are not always that useful, but there are some research going on at the moment to try and sort of figure things out about this. This is still very new technology, and we don't completely understand everything there is about it. It's still very much an area of research. So we don't know exactly how to turn on some of these genes or turn them off in terms of growing specific cells. That comes with specific risks of its own. So a stem cell, the whole point of a stem cell is that it grows rapidly. One other condition to be aware of with that is cells that grow rapidly in the body have the ability to form cancers and tumours, and we've talked about that previously. So there are concerns that actually these rapid growing stem cells may not be controlled and potentially could lead to the patient gaining cancer or a tumour of some form. The other is that if we are taking stem cells from an adult, there is the potential for them to be carrying viruses. So if we are taking cells from one individual and giving them to the other, potentially we have got viruses or disease carried within those cells that we're not aware of. And so you've got the potential for causing issues associated with that. Obviously, there is the positive aspect that you are having the potential to cure illness and disease. And so that does have a positive aspect and a positive value as well. But you can see it's quite a complex argument, and in an exam question you're going to need to be prepared to argue both the positives and the negatives. Okay, let's have a look at some practice questions then, just as we've done previously. You have 3 questions on the screen there, each worth 1 mark, so you need to give yourself a maximum of 3 marks to try and do this. Remember to read very carefully. It's asking you to tick 1 box only for each question. So pause the video here, have a go, and then we'll go through some answers in a moment. Okay, let's have a look then. So what type of cell division happens to produce new body cells? So new body cells are coming from mitosis. Meiosis is sex cells, so that is literally thinking about sperms and eggs and creating baby, and differentiation is the process that happens when cells become specialised for a specific purpose. Part 2 says, why can cancers grow very large? Okay, so we've got cancer cells are specialised, cell division is slow. Well, we know cell division is not particularly slow in cancer cells. It is uncontrolled, and the growth happens very quickly, forming those cancers and those tumours. Right, and finally, give 1 factor which increases the risk of getting cancer. Lots of different things you could put here. Smoking is probably quite an obvious one that you could come up with. You could be thinking about UV light in terms of skin cancers. You could name a specific chemical that is cancer causing, or generally something that we call a carcinogen. Okay, lots of different options there that you could be thinking about. Okay, so the next one then, 5 marks. Describe what happens in each of the 3 stages of the cell cycle. Okay, so 5 marks for this one. So 5 points you need to be thinking about as you explain the stages. Okay, so I'm going to talk through the whole of the cell cycle process, and I'm going to make more than 5 marks for more than 5 points here. And any of these 5 would get you the marks. Okay, so the first point would be to state what happens at each stage. So stage 1, we see the cell growing, so we see cell growth. And we see an increase in the number of organelles. Okay. We also, at this point, see the DNA replication happening. Okay, so everything is doubling up ready. Stage 2. Stage 2, 1 set of each of those chromosomes moves to each end of the cell. Okay, and at this point as well in stage 2, we've got the nucleus dividing. And then we're on to stage 3, where we see the cytoplasm, oh, missed an S, there we go, and the cell membrane and everything else beginning to separate and divide. And we are left with 2, important you say this, genetically identical cells. Okay, so you can see I've made a number of points there. It might be that for stage 1 all you do is say that cell growth occurs and then you say a lot more detail for the other stages. As long as you have made 5 individual points and you have mentioned what happens at each of the 3 stages, you would have got the 5 marks there. Right, here we go then, the last question we're going to have a look at today. So figure 10 shows how embryonic stem cells are produced in therapeutic cloning use in patients. Give 2 advantages and 2 disadvantages for 4 marks. Off you go. Okay, now there are a huge range of answers that you could have put here. So let's look at a few of the options. So I'm going to put my advantages up on this side and then I'll put my disadvantages over on this side so I can give you a range of options. So the first thing you might have thought about is this ability to cure disease. Okay, so it's the fact it can cure the disease or the medical condition. It might be that you've gone down the route of what it's providing you with. So you're able to produce cells of any type. You might be thinking about the fact it's specifically this therapeutic cloning. So we know that we've got these genetically identical to the patient cells. So they are unlikely to be rejected. By the patient. Okay, so that's quite an important point there. Could be the fact that you can produce many cells. Could be that you're producing a new organ, so perhaps this would reduce the waiting time for an organ transplant. Okay, I'm sure there are loads more there that I haven't even thought of, but there's a range of possibilities that you could have put there. I think the most likely ones that you are going to come up with based on what we've talked about are the fact that it is going to be curing a disease and that the cells are unlikely to be rejected. So I think those would probably be the 2 most obvious ones that I would see there. And then the disadvantages then, what's wrong with it? Well, we've got our ethical and our religious arguments, haven't we, that we need to think about, but we need to be specific. It's no good just putting ethical reason, unethical or something like that. We need to be a little bit more specific. So we've got the fact that it is a potential life that is being killed or destroyed. We've got the fact that there are risks we don't know about. So such as this idea of cancers from this uncontrolled growth. We've got as well the fact that we may transfer a virus. Remember, viruses could be present within these cells that we're replicating. Could also be thinking about the amount of embryos that have to be created, the number of embryos created that fail, that don't survive to be usable for this. Could be thinking as well, these embryos, as you can see up here, look, require a donated egg cell. So it might be that there isn't enough of those egg cells. Okay, so I think the ones you're most likely to have thought of here is probably the fact that we've got the life being destroyed, and it's probably one of these risk factors here, whether it be cancer or transport of viruses. Okay, So that gives you a range of options to have got those 4 marks. And that is the last question for today's session. So thank you very, very much for all of your efforts and all of your hard work, and good luck in the assessment session next time.